Hello everyone, my name is Apisala Pipat. Today I'm gonna present about anti-cancer effects of combined zoledronic acid and doxorubicin in triple-negative breast cancer cells. Advisor is Assistant Professor Vanarasamy Getchat, and this is academic year 2022. This is the outline of this presentation. In 2017, number of deaths caused by cancer reached 9.56 million, which is the second place after cardiovascular disease. Focusing on cancer in women, breast cancer is in the first place of number of cases and in the first place of leading cause of death compared to all cancer found in women. This is the trend of age-adjusted cancer death rate in women from 1930 to 2018. You can see that breast cancer has the highest mortality rate, and after 1989, the mortality rate is declined due to early detection and prompt treatment. Signs and symptoms of breast cancer. Most patients has no symptoms, but symptoms may include lump, mass, irritation, swelling, pain in breast or nipples, on nipple discharge, nipple retraction, and nipple scaling. Breast cancer can be divided into 4 molecular subtypes: luminal A, luminal B, HER2, and triple-negative breast cancer. Luminal A has ER and ER positive, while HER2 negative. It has high expression of ER-related genes and low expression of proliferation-related genes. Luminal B has ER positive, and if PR positive, PR is positive, but if PR is negative, HER2 can be positive or negative. It has high expression of proliferation-related genes. HER2 subtype has ER and PR negative, while HER2 positive, and triple-negative breast cancer has ER, PR, and HER2 negative, but it can have some protein expression such as CK56, CK14, 17, p-cadherin, and EGFR positive. The treatment can be divided into local treatment and systemic treatment. In local treatment, there are surgery and radiotherapy. While systemic treatment, for example, hormonal therapy, HER2-targeted therapy, and chemotherapy. Systemic treatment recommendation according to molecular subtypes. Luminal A use hormonal therapy due to the positive of ER and PR receptor. In luminal B, we use chemotherapy followed by hormonal therapy. And in case if it has the HER2 receptor positive, we use anti-HER2 therapy as well. In HER2 positive subtype, use chemotherapy and anti-HER2 therapy. And TNBC use chemotherapy. Survival rate in each subtype. It differs between each subtype. The luminal A has higher survival rate, followed by luminal B, HER2, and triple-negative breast cancer has the lowest survival rate. So we are interested in TNBC. Triple-negative breast cancer, or TNBC, is the most aggressive and worst prognosis form of breast cancer. It has high heterogeneity and has no specific pathway due to ER, PR, and HER2 negative, so we cannot use the targeted therapy. So the main treatment is chemotherapy. Tumorigenesis of TNBC. The first one is receptor tyrosine kinase and downstreaming signaling pathway, MAP kinase pathway, and PI3K/AKT pathway. This is the MAP kinase pathway. Selumetinib or is a MAC inhibitor. It inhibits migration and invasion in MB231, which is TNBC cell lines. PI3K and AKT pathway. mTOR inhibitors can resensitize platinum-based therapy resistance, such as cisplatin, and PI3 mutation and PTEN loss activity was found in TNBC 23.7% and 25 to 30% respectively. The PI3CA mutation and PTEN loss activity can cause hyperactivation of PI3K/AKT pathway, resulting in cell proliferation and cell differentiation. Second mechanism we're going to talk about is the epithelial to mesenchymal transition, and when beta-catenin pathway leads to cell invasion. This is the epithelial to mesenchymal transition, or EMT. On the left-hand side, you can see epithelial cells, and epithelial markers are cytokeratin and E-cadherin, for instance. When epithelial cells start to transition from epithelial cells, and it can change into mesenchymal cells, which is prone to cell invasion. Mesenchymal cell markers are vimentin and cadherin and fibronectin and etc. We found that EMT are more frequently expressed in TNBC than other subtypes, which play roles in tumor invasion and metastasis. This is the Wnt beta-catenin pathway. Beta-catenin acts as a co-transcriptor of tumor oncogenes, and it is significantly increased in TNBC patients compared to other non-TNBC. Decreasing of beta-catenin can lead to reduction of proliferation and metastasis. Another study suggests that when stimulates the migratory ability of MDAMB 231. The last one is eukaryotic translation initiation factor, or EIF. EIF is the function of EIF is to initiate the translation, thus have the function of protein synthesis. The abnormal function of EIF can cause abnormal translation, resulting in abnormal gene expression. Among all the EIF, EIF3 is the most complex form, and EIF3 composing of many EIF subunits. One of that is EIF3M. EIF3M is responsible for stabilizing the core subunits. Therefore, the depletion of the EIF3M can result in marked decrease of translation and cancer cell proliferation. EIF3M has higher expression in TNBC than others, so the upregulation of EIF3M in TNBC is correlated with lower survival rate and relapse-free. Next, we're gonna talk about the chemotherapy, or doxorubicin, which is one of the main first-line treatment chemotherapy in TNBC. When doxorubicin enters the nucleus, it interferes with DNA and also inhibits topoisomerase 2 alpha, which is an enzyme in DNA replication. Doxorubicin in cytosol can cause a reactive oxygen species and react to cause lipid peroxidation and cause membrane damage. ROS also generate oxidative stress, causing cell death. However, we found that doxorubicin can have many resistant mechanisms. The first one is TOP2A amplification. As I mentioned earlier, doxorubicin, one of the mechanisms of doxorubicin is to inhibit TOP2A. When TOP2A is amplification, doxorubicin can relatively inhibit TOP2A less than it should be, so it progresses into cell proliferation. The next one is ABC transporter regulation. This is a P-glycoprotein, which is one of the ABC transporter in ABCB family. The function of P-glycoprotein is to efflux doxorubicin outside the cells. So the upregulation of P-glycoprotein resulting in the more efflux doxorubicin outside the cell, resulting in less doxorubicin inside the cells. Next is PD-L1 expression, which is found in 20% of TNBC patients. When tumor cells express PD-L1, T cells, which is an immune cell, recognize tumor cells as normal cells. Thus, the T cell will not kill the tumor cells. Next one is hypoxia. Breast cancer is considered as one of the solid tumors. So the cancer cell, when it is proliferates, there are some malignant cells that are distant to the blood vessels, resulting in insufficient oxygen supply called hypoxia. The prolonged hypoxia or chronic hypoxia is known to be firmly correlated with drug resistance due to various reasons. For instance, poor drug penetration, acidic tumor environment, and also ineffective of cytotoxic effects of doxorubicin due to its oxygen-dependent process or generating the reactive oxygen species. And the last persistent mechanism is activating an F-kappa B pathway. It is known that DNA damage that caused by doxorubicin can activate an F-kappa B pathway, which can cause cell proliferations. So TNBC encountered many drug-resistant problems through several mechanisms, as we talked earlier. And also we have the problems about its adverse effect, which is cardiomyopathy. So the researcher aim to find new substance to overcome drug resistance and improve safety of treatment. And that substance is zoledronic acid. Zoledronic acid is an anti-resorptive drug in amino bisphosphonate group. The mechanism of zoledronic is to block osteoclast activities and induces osteoclast apoptosis, resulting in decrease of bone resorption. Indication of zoledronic acid is hypercalcemia in malignancy and patient with multiple myeloma with documented bone metastasis from solid tumors. Recent research have found that zoledronic acid has potential anti-cancer effects. The first one is to increase apoptotic activity and inhibition of cell proliferation. About the induced apoptosis, we found that zoledronic can activate the pro-apoptotic proteins caspase 3, 8, and 9 in oral carcinoma. It increased cleave-pop in oral carcinoma and reduced anti-apoptotic proteins, including BCL1, BCL2, and BIT expression in oral carcinoma. About the decreased proliferations, zoledronic acid when combined with cisplatin decreased BCL1, PMTOR, and MAP kinase in MDA-MB231. It also inhibited the topoisomerase 2 alpha activity in mesothelioma, decreased cyclin E1, B, D1, D3, CDK1, CDK4 in MDA-MB436 in immunocompromised mice, and it also arrests cell cycle between S phase and G2M phase in fibrosarcoma and non-small cell lung cancer. The next effect is anti-angiogenic effects. Zoledronic acid can reduce VEGF mRNA and protein expression and VEGF secretion in non-small cell lung cancer, resulting in inhibits of endothelial cell proliferations. It also increases intracellular IgG, which can convert it into APPPL, which has inhibitory effects of revascularization. And the last effect we're going to talk about is decreased tumor invasion and migration. It reduces FcR4, reduce MMP9, and inhibit mesenchymal stem cells activation. It also suppresses Wnt protein at mRNA level in prostate cancer and synosine RNA in FCF7 and SKBR3. So overall, breast cancer is the most common cancer in women worldwide and has the highest rate of death. Among all of the breast cancer, TNBC has the worst prognosis. And so the treatment of TNBC is chemotherapy. The first line chemotherapy, including doxorubicin. However, TNBC encountered drug-resistant problems and it adverse effects, so we aim to find new substance to overcome drug resistance and improve safety of treatment. And that substance is zoledronic acid. The rationale of the study, why we combine zoledronic acid in MDA-MB231, because the first is zoledronic has anti-proliferative effects, anti-migration and invasion effects, and anti-angiogenic effects in many cancer cells, such as non-small cell lung cancer, fibrosarcoma, oral carcinoma, mesothelioma, and also other breast cancer cell lines. And also, there's no previous study that show combination effects in varying concentration of zoledronic acid and doxorubicin in MDA-MB231, and discuss about the specific molecular targets, whether it's the combination effect is an additive or synergistic effects. And there's no previous study that showed the effect of zoledronic acid in topoisomerase 2 alpha in MDA-MB231. So we think that the benefit from combining zoledronic acid with doxorubicin may decrease doxorubicin resistance in MDA-MB231 and decrease doxorubicin dose, resulting in decreasing of adverse effects. The objective of this study. The first is to study the effects of zoledronic acid on the inhibition of cell proliferation, angiogenesis, and cell invasion migration in TNBC. Second, to study the effects of zoledronic acid combined with doxorubicin in TNBC. And the third one is to investigate the mechanisms of action of zoledronic acid in TNBC. Research questions. First, does zoledronic acid exhibit anti-proliferative effects, anti-angiogenic effects, and anti-invasive migration effect in TNBC? Second, does zoledronic acid combined with doxorubicin show increased effect in triple-negative breast cancer cells? And third, does zoledronic acid inhibit proliferative factor, which are cyclin D1 and TOP2A, angiogenic factor, which is VEGF, and invasive migration factor, which is CXCR4 in TNBC. The hypothesis of this study. First one is zoledronic exhibit anti-proliferative effects and anti-angiogenesis and anti-invasive migration effects in triple-negative breast cancer cells. Second, zoledronic acid combined with doxorubicin shows increased effect in triple-negative breast cancer cells. And third, zoledronic acid can inhibit proliferative factors, angiogenic factors, and invasive migration factors in TNBC. The conceptual framework, we divided TNBC into three parts. The first part is cell proliferation. We studied on TOP2A and cyclin D1. Angiogenesis, we focus on VEGF, and cell invasion migration, we focus on CXCR4. And we use the doxorubicin, zoledronic acid, or they combine and inhibit all those parts. The experimental designs about the zoledronic acid alone, cell proliferation, the cell viability, we use MTT assay, and the proliferation factors, angiogenic factors, invasive migration factors, we use the RT-PCR and Western blot. And about the cell invasion migration, we use the scratch assays. And in doxorubicin and combined ZA with doxorubicin, both sequential and non-sequential treatment is as we talk about the zoledronic acid alone. Scope and delimitation. We have the in vitro study using triple-negative breast cancer, which is MDA-MB231 cell lines. Research study. The research design is experimental study, which is an in vitro study. This is the data and statistic analysis we use in the study and ethical consideration that will be exempted from IRB. These are cell lines, chemical substance, and materials. Research method. The first one is cell preparation. We're gonna culture cell in cell culture dish, and the media we used is the membrane glucose, 5% of FBS, 1% of penstrep, 1% of L-glutamine, and 0.1% of amphotericin. Incubated the dish in 37 degrees Celsius and 5% carbon dioxide. About the MTT assay, that study on cell proliferation, after we seed cell in the 96-well plate, we will be waiting for 24 hours and then treat with zoledronic acid or doxorubicin all combined as the timing we showed above and concentration. Then we add MTT and remove media. Then we measure the absorbance of the formation product, then calculate their viability and IC50. Next is the study of cell migration by scratch assay. After we seeding cell into 6-well plate and wait for 24 hours, we scratch each well, then treat it with zoledronic acid or doxorubicin or both. Then we observe the scratch as timing follow in the slides, and the concentration of zoledronic acid and doxorubicin will be non-toxic concentration, and combined will be non-toxic concentration as well. Then we measure the cell surface area, then calculate the percent migration. Next is study of cell invasion by matrigel invasion. The upper chamber we put MDA-MB231 cell line, zoledronic acid, and doxorubicin and serum-free media. While in the lower chamber we put the serum media. After we put, we waiting for 72 hours and then fix with formaldehyde and stain with crystal violet. Then we count the cells under light microscope. 5-year study of gene and protein expression involved in cell proliferation, angiogenesis, and invasion migration using RT-PCR and Western blot. The study of gene expression we use RT-PCR and protein expression we use Western blot. The gene and protein we are interested is cyclin D1, TOP2A, VEGF, and CXCR4. Study of gene expression using RT-PCR. After we seed cell, then we treat with drugs. And we extracted RNA after 72 hours with triazole reagent. Then we measure RNA concentration at 260 nanometers. Then we converted RNA back to cDNA using reverse transcriptase, then amplify using RT-PCR. Next is the study of protein expression using Western blot. After we prepare the sample, we use the cell gel electrophoresis and transfer into membrane. Then we block with milk and incubate with primary antibody, then secondary antibody, then detection and analysis. Expected benefit and application. This research will study on the effects of zoledronic acid combined with doxorubicin. So we expect that this research can lead to further understanding in potency and mechanism of resistance of breast cancer, suggest another promising way to treatment for patients with previous drug of choice resistance and improve patients' quality of life. This is the time schedule and administration. The budget. Thank you. This is the take-home message. In invasion and migration assay, why do we treat cells with non-toxic concentration? Thank you.